Hello to our friends joining us via recording. Today we are working on week 3 in lab. So in week 3, we're covering all of the cell cycle worksheet. That's lesson number 3, and the first part of lesson number 4, histology. Before we dive into those worksheets, I want to mention something about the Wiki Project. My screen is going to update here to show you what it looks like when you are in your group. Let me see if I can make it a little bigger. When you're in your group to work on the wiki, remember that down here at the bottom in our group tools, we go into our wiki. The first wiki assignment was due this last Sunday. So this first page that you land on, part 1, your whole group should have already done. Here's what I want to point out to you though. Over here on the right, notice how underneath it, there's a thing that says Lab Group Wiki Project Part 2. When I click on this, it takes me to a screen that now has the second part of this project. The second part of this project is due by this coming Sunday. So the 7th, 4 days from now, part 2 of this project is due. When you go into your group's wiki, you're going to do just like we did last time, where you put your name on a line. Now, when you put your name on that line, so say I was part of this group and I put my name here on this top line, notice that it says I'm student number 1. Maybe you go into your group wiki after class today and you notice that already 2 of your classmates have put their names on the line. So you're down here on this third line here, you're student number 3. So whatever order you end up putting your name on, whatever line your name is on, that tells you your student number. If you're 1, 2, 3, or 4. That matters because when we go down into this activity, you're going to be measuring different types of cells. To make sure that everyone in the group doesn't measure the same thing, you need to use that student number. So if you're the first person in your group to sign up, you're student number 1. If you're the second person, you're student number 2, number 3, or number 4. So this way, all of these types of cells get measured. Everybody measures a different set of cells. So the question I had in the email was, how do you know your student number? It's whatever line you end up signing up on. So sign up on the first available line. That gives you your number. When you know your number, you'll click here. Let me see if it's going to work for me. When you click here, this is going to take you to a slideshow that's hosted online. This slideshow has each of the types of tissue in it that you're going to be measuring. So the second part of the Wiki project, we're measuring different types of cells to see if all the cells are the same size or if they're different sizes. So notice, if I was student number 1, I'm going to be measuring simple columnar epithelium. And I'm going to be measuring adipose connective tissue if I'm student number 1. If I'm student number 2 in my group, I'm measuring erythrocytes, which are red blood cells, and I'm measuring elastic cartilage cells, chondrocytes. For you to be able to measure, you're going to need to download this slideshow onto your computer. So see this little thing right here, this download arrow? If you click on this, it's going to download the slideshow onto your computer. Let's see how my computer does. Okay, so you download it on your computer. You're going to be able, once you have this downloaded on your computer, you're going to be able to move the little ruler that's on each of these slides to put it next to cells to figure out their size. I probably should have done this before class. I apologize. Here we go. So I'm going to enable editing. Here's what it looks like when you download it onto your computer. So say I'm student number 1. I can take this little ruler right here and I can move it next to a cell. And I'm going to, so the ruler, notice I have a note here that this whole ruler is 10 micrometers. So I'm going to look at, with it on my picture here, I'm going to estimate about how tall this ruler is. Notice if I decided to pick a cell that was really slanted, see how this one's kind of slanted? There's a green dot right here that I can use to rotate my ruler. and then I can measure it a little bit more effectively. So the ruler moves to help you go through and find the cell you're looking for. It's labeled multiple places, but please don't resize anything. So don't click on this part. Here, let me show you what not to do. This is what not to do. Oops, do not want to do that. We don't want to make that ruler a different size or we'll measure ourselves wrong. The other thing we don't want to do is change the size of these tissue pictures either. It's going to be harder for you to do that, but if you accidentally click on it and we start dragging stuff over here, that's going to make the size of those tissues change. So please don't do that. Please don't change the size of the ruler. Just move your ruler over, measure what it looks like the cell. You're going to measure 3 different cells. So maybe I want to do this one. I'll measure how tall this one is. Maybe I want to go up here and measure this one. Notice I can rotate my ruler again so I can find the size. So pick 3 different cells that you see on your tissue. Again, the ruler moves. I can take my ruler right here and I can drag that onto my picture. I can measure this one. I can measure this one. I can measure this one just for a little bit of variety. So for each of these types of tissues, look at the number. If you're student number 2, you ignore those first 2. You just do this one and you do this one. If I'm student number 4, I'm going to go all the way here to the end. I'm going to measure these cells that have little arrows pointing to them. So pay attention when you're on each of these slides. What student number is doing this type of tissue? And the only thing that moves here is the ruler. Record the size of 3 different cells once you've measured it, and that's what you're going to put into your group wiki. There was a comment in the chat that's totally correct. Say I accidentally make my ruler big. Oh no, I'm freaking out. I resized my ruler. You can either hit the Control button and Z on your computer to make it undo what you just did. Or if you go up here at the very top, see next to the Save button, there's this undo arrow. If I click that, that'll get me back to. So if you happen to change the size on your ruler, undo it, either using this little back button or using Control Z. That'll get you back to the right size. If you really mess things up and you feel like you can't get the ruler back to the right size, just download the PowerPoint again. It'll go back to the right size for you. So this is the document that you'll use to actually measure the size of your cells. Pick 3 different cells that you see on your type of tissues. Again, reference your student number, which is the order that you sign up in your wiki, to know which cells you're measuring. Okay, so there's my cells. We did our measuring. Now we'll come through on the wiki page. You're going to record your name, and you're going to record the size of each of those 3 cells that you measured. Then we're going to make you do just a little bit of math. You're going to calculate the average size of the cells that you were looking at. So reminder from your math teacher that the average, we would add the size of each of those cells that we measured together and then divide it by 3. Whatever number you get, we're going to put here in the average column. So you're measuring the size of 2 types of tissue, the cells in 2 types. So if you're student number 1, you only fill in these first 2 rows. If you're student number 2, you just fill in these 2 rows, number 3 and number 4. You're only responsible for measuring 2 types of tissue. Then everyone in your group has to measure or has to answer this question down here at the bottom. I'm half reading the question, half talking. It's not working for me. So everybody in your group, notice there's a space for each of you to answer this question about the size of tissues. So make sure that you not only fill in the table, you also fill in your answer to this question down here below. I know I saw something. Let me pull up my chat to see. How do we write the unit for measurement on the table? That's a great question. You can put it in, let me type it in the chat, as micrometers. Here, let me check. I don't know if they actually have that micron symbol. I'm going to pull up the edit to see if it gives me the option. When I am on the table. Let's see. I'm going to try symbol. We'll check together. It looks like a funky, oh yeah, here it is right here. So you can either put, so see this little guy right here? This is micro right here. You can either use this. Let me remind you how I got there again. I got there using this little C, the copyright symbol. So I can type in my number, let's say it was 10. I put in that symbol and put an M after it, so micrometers. Or I'm not gonna get mad at you if you just use the letter U. That's a lot faster and easier. So if you just wanna type UM, I'm cool with that too. So you'll go through and you'll fill in your parts of the table here. So then we'll say this one, we'll make this up, it's 6.5. Then I would calculate what the average is, whatever my average is. Again, I just made that up. I'm done with one type of tissue. And then I'll do that with my next type of tissue. Remember, just like on the other wiki, once you've typed your things in here, we want to make sure that you go back to your color. So there's my letter T for my color. Let's say my color was blue. I want to make sure that everything I put in, every answer I write, is in my color. So there's my color there. Somebody mentioned they're having trouble accessing the PowerPoint. Most likely what that means is that you have a personal Gmail account that is logged in. You need to be logged into your MyTCC Gmail account to access that slideshow. So try logging out of your personal Gmail account and try clicking it again. That should let you in. The other place you can access it as well, if you have trouble using that link, so if you have any trouble when you're trying to complete this, if clicking here is not working, you can also go to the student Google Drive. The student Google Drive is in the lab resources area. So let me show you the other way to get to that PowerPoint. Lab resources. If I go to start here, the student Google Drive is down here at the bottom. So if you click on this, it will take you into that Google Drive, and that's another way to get to that PowerPoint. Either way, it's exactly the same. So use whatever works. Edith says we're in business with her too. Excellent. So that's great. Any other questions about the wiki before I move on from it? Let me see if I can get it back. Yeah, so Fanshin, if you're having issues, again, I'd say try going through the Google Drive directly. And you are probably going to have to download it onto your computer. So if it's having trouble downloading it because it's too large, maybe try to find another computer that you could use. Maybe that you don't have space for it. That would be my only other thought. So Dennis, the place that you'd go to find it is here, Group Wiki Projects. So one more time, when you're in Blackboard, Group Wiki Projects, you'll select your lab group. I've got a million groups because I can see everyone's. So you'll click on your lab group. When you're in your lab groups page, remember that the wikis are down at the bottom. We'll go into our group's wiki. And now we're working on part 2. So it defaults to part 1. We want to change it to part 2 by clicking over here. And that will load what we were just looking at. All right. As you start working on this project, please feel free to reach out with any questions that you have. I want to make sure we have plenty of time during today's class to cover some of our other content, but I'm happy to troubleshoot it with you if you continue to have any trouble. So feel free to email me. Also, I'm available Friday afternoon from 1 to 2:30. Remember, that's just kind of a general Come with any questions that you have time. So feel free to stop by Friday as well. I'd be happy to help you with it then too. Before we started our discussion of the wiki, before we started our recording, we did a little bit of group work to help us with remembering some of the most important stuff about the cells in the body. We're going to be talking about cells dividing, and we're actually going to use some of the parts of a cell as markers for us to tell whether or not that cell is dividing or not. So one of the things when we're talking about cells dividing that we're always looking for on our cells is we're always looking for the location, or we're always looking for their plasma membrane. When a cell goes from being 1 to dividing into 2, we take our cell membrane or our plasma membrane, which I'm sure we found in our groups was this green line here on the outside. We actually take this plasma membrane on the outside of the cell and we kind of pinch it in the middle to split up the cell into 2 different parts. So the plasma membrane of the cell located on the very outside, you will know that the process of cell division, which is called mitosis and cytokinesis, we will know that that's done when I see 2 separate plasma membranes. Help me out in the chat. When we're talking about the plasma membrane, how would we describe its function? What's the job of the plasma membrane of a cell? It's kind of multiple jobs, actually. What are some of those jobs of the plasma membrane? Yeah, perfect. So there's kind of a couple of ways that we can think about the jobs of the plasma membrane, and we're hitting both of them there in the chat. The first way that we can think about the job of the plasma membrane in the most simple terms is it divides the inside from the outside. If we want to think just biggest picture, what's the job of the plasma membrane? Divides the inside from the outside. But like several of us mentioned, the reason it's important for us to divide up the inside from the outside is because the fluid that's inside the cell is different from the fluid that's outside the cell. So we could be even more specific, and we could say that we divide intracellular fluid, see if I can spell that, intracellular fluid from extracellular fluid. If we want to be a little more specific about what the plasma membrane does, divides the fluid on the inside from the fluid on the outside. Hey, when we talk about that fluid on the inside of a cell, there's only one name for it. What's the name of that fluid inside a cell? The fluid inside a cell. Yeah, absolutely right. Lots of us mentioned that fluid is called cytosol. Absolutely. So the plasma membrane's job, keep the cytosol inside, keep the extracellular fluid, which has lots of different names. We won't list them all, but keep the cytosol separate from those fluids that are on the outside. Keep the inside of the cell different from the outside of the cell. And during tomorrow's lecture class, we're going to talk about how chemistry makes that happen. So, function of the plasma membrane, divide the inside from the outside. Excellent. I also asked you in your groups to identify where the centrioles were. It's going to get a little bit crazy here, but go ahead and go up here above my pointer, grab that pencil. Let's circle those centrioles really fast. Can you show me where the centrioles are in our cell? Awesome. Yep, we found them. So if you recall from class last time, lab class last time, I told you, I'm going to clear it off so we can see them again. I told you that the centrioles kind of look like churros. So we got a couple little churros going on in the cell here. When we talk about the churros that are the centrioles, Their job is going to be to divide the genetic information in your cell when it's dividing. So they're really important for pulling apart our genetic information when it's time for the cell to divide. So centrioles right here, my churros. If I flip over to my other cell model, we also see them right here. When we see them over here, they maybe look a little bit more like licorice. Yeah, some are mentioned on the other slide. They kind of look like breadsticks too. Yeah, I'm all for food analogies, just saying. Anything that's going to help you remember what the centrioles look like, some kind of food memory, or basically they come in a little pair and they float around in the cell. So here's my centrioles on this slide. We saw those centrioles on the other one as well. One part of the cell that's going to be really important for us to pay attention to when we're talking about where we're at in the process of cell division is that nucleus. So the nucleus, I'll point out for us, is this part right here. So this light purple, everything that's inside of it is part of the nucleus. This fuzzy black stuff is the molecule that I mentioned I wanted you to tell me was what was stored inside the nucleus. What do we find inside the nucleus? What's this fuzzy black stuff here? Yeah, exactly. So this fuzzy black stuff that we see right here is the DNA, your genetic information stored inside the nucleus of the cell. Yeah, so Elapina's totally right. Today during our lab, we're gonna be more specific about this particular form of the DNA. This loose form that kind of looks fuzzy, like we see in this picture, is something called chromatin. So when your cell is just chilling out doing its normal everyday life, it leaves your DNA in this really loose, I sometimes call it like a spaghetti noodle mess called chromatin, where it's just all mixed up with each other. Chromatin's really great when you need to use that DNA to build a protein. Chromatin is not really great when you're trying to equally divide your genetic information. So when it's time for us to divide our genetic information, we're going from one cell to another, we'll take this spaghetti noodle mess that is chromatin and we'll condense it down into those X-shaped structures that you're used to seeing. What do you call the X-shaped form of DNA? Does anyone remember what we call that form of DNA? Yeah, exactly. So this form of DNA is called a chromosome. Normally, when you were in your high school biology class, when we think about DNA in general, you usually see it in this X shape. But the X shape, the only time we have that is when the cell is dividing. Because it's really easy for me to snap this X in half and equally divide the genetic information, but I can't really use the genetic information that's in this chromosome. Like I can't make proteins from it. So the only time a cell is going to have the X-shaped chromosomes is when we're actually dividing it. Normally, we're going to have this loose form called chromatin, the spaghetti noodle mess. I had a colleague that described it kind of like ramen noodles. Again, we do love those food analogies, right? So whatever helps you remember that here inside the nucleus, we've got DNA that's loose and kind of puffy. That's the chromatin. When it's time to divide, we'll turn that puffy stuff into chromosomes. Chromosomes, that X-shaped structure. There were two organelles that we talked about last week that helped us to make proteins. What was the name of the two organelles that were involved in protein synthesis? We had two of them. Yeah, so the first one, which is the large, I'll emphasize here, large organelle, that one is called the rough ER, the rough ER. It's called rough because of the small organelle that is attached to it, and those are the little things called ribosomes. Ribosomes. So again, when we're looking at our cell, ribosomes are these little red dots that we see here, free floating, or we also see them all along on our rough ER here. So all those little red dots on here are ribosomes. All of the places where these flat purple membranes are covered in red dots, that's the rough ER. What was the name that I used for this part right here? The purple parts that didn't have ribosomes? What were these things called? See them kind of all over the place on this model here. Yeah, so these ones that don't have those red dots were the smooth ER. The smooth ER looks smooth, no ribosomes attached to it. That's why it's the smooth ER. The rough ER looks rough with all of those ribosomes. Yeah, Kaylin, I like your comment. You're good. Kaylin said, I meant smooth ER, not smoother. But you're not wrong. It is smoother, right? Perfect. Okay, so when we do cell division, which we'll talk about shortly, we're going to talk about a group of proteins that helps us to divide up our chromosomes, to divide up our genetic information. Remember, keep in the back of your mind that those proteins are either made by the rough ER or they're made by the ribosomes. That's going to help our cell with division. Let's switch gears just a bit from talking about cells dividing to talking about the 4 types of tissue in the body, because a couple of them have a whole bunch of proteins. So let me pull up a picture here, and we're going to our lab packet for lesson number 4. Lesson number 4, lab packet. Is this the first page there of the histology lab packet? Do we have these pictures? I think we do, right? Okay, we do have those pictures. We are in the lesson number 4 packet. Lesson number 4, let me give you some terminology here. Lesson number 4 is called histology. Histology. Histology is the study of tissues. Study of tissues. And when we talk about tissues, we don't actually mean the things that you blow your nose with, but if it helps you remember, go for it. Histology, we're studying tissues. What tissues are is when more than one cell works together in your body to do something. So when we talk about tissues, when we talk about histology, all of the types of tissue that you have in your body fall into 4 different groups. So there's only 4 kinds, big picture, 4 kinds of tissue in the body. Let's talk about each of these kinds of tissues and what makes them different from each other. I'm actually going to start in a weird place. I'm going to start right here with this type of tissue. Can you help me out in the chat? Do we know what kind of tissue we're looking at right here? What kind of tissue is this one? Yeah, so this one down here, as several of us have mentioned, this is an example of muscle tissue. Muscle tissue. The reason I wanted to start with muscle tissue is because all of these stripes that you see here inside this cell, all of those stripes are because of the many proteins that these cells have inside of them. So this is, like someone mentioned, this is specifically skeletal muscle tissue. So the kind that helps you to move your skeleton. These cells in skeletal muscle tissue are completely filled with proteins. So lots and lots of proteins inside these cells. These proteins are going to be important for a few different things in your body. When we talk about muscle tissue, the first thing that muscle tissue helps us to do is movement. So this could be movement of your entire body. This could also be movement of things like food through the digestive system, or blood through your arteries, or movement of blood through the heart. So first thing we should be thinking with muscle tissue, this is my movement tissue. Second thing we should think with muscle tissue is that it helps us to generate heat. This is why when you're cold, you shiver to make some heat. Also, your basic body temperature, a lot of that comes from the fact that your muscles contract. So muscle tissue helps you to move. Muscle tissue keeps you warm. And the other thing that muscle tissue does for you is it gives you your posture. Now, this whole quarantine stay-at-home thing has really messed with our posture, right? Because our muscle tissue maybe hasn't quite been doing what it should be doing. So these are some of our main functions of muscle tissue. And again, muscle tissue, when we look at it, the cells are completely filled with proteins. It's those proteins that help us to do things like movement or doing your posture, giving you heat. Another type of tissue that has a lot of proteins in it is this one up here. Can you help me out in the chat? Which kind of tissue are we looking at in this picture? What kind of tissue is up there? Yeah, exactly. Up there, we are looking at connective tissue. Oops, that's not going to fit. Connective tissue. Now, we are going to look in a couple of weeks at all the specific kinds of connective tissue. So that's coming soon. Actually, it might be next week. Coming soon. Here's my wink, wink, nudge, nudge for you. When we look at a type of tissue, if you're having a really hard time figuring out which of the 4 main types of tissue we're looking at, your best guess or your safest guess is to go with connective tissue, because connective tissue is kind of like the catch-all category for every weird type of tissue. So anything from the kind of connective tissue that we see in this picture, which is absolutely, it's called areolar loose connective tissue, to the type of tissue that we read about in lecture, adipose tissue with fat cells. That's another type of connective tissue. Your blood falls into the category of connective tissue. Bones fall into this category. So connective tissue looks really different depending on which kind of connective tissue you're looking at. But here's some of the things for us to know about connective tissue. Connective tissue has something called extracellular matrix. Extracellular matrix. We know what that word extracellular means. What does that word extracellular mean? Do we remember? Extracellular. Yeah, several of us are mentioning. It's just like that extraterrestrial that we talked about, someone that's outside the Earth, like ET. Extracellular matrix just means stuff outside the cell. Stuff outside the cell. When we talk about connective tissue, the stuff outside the cell is things like these dark lines that are proteins that we see here. It can be things like when we talk about bone tissue, we've got a lot of calcium outside of the cell. Connective tissue has a bunch of stuff on the outside of its cells. That stuff is called extracellular matrix. It's hard for us to say one particular function of connective tissue, except to maybe say that it connects parts of the body together. If we want to think of a big picture thing. So connective tissue is going to connect your muscles to your bones. It's going to connect your bones to each other. We're going to use it to transport oxygen throughout the body. So it's kind of connecting your lungs to your organs in that regard. So connective tissue, just kind of a big picture, we do everything. We connect it all together. Lots of extracellular matrix. Let's compare the fact that connective tissue has a bunch of stuff on the outside to my type of tissue over here that doesn't have anything on the outside of the cells because it's made entirely of cells. Which of the 4 main types of tissue do we see over here? Who's this over here on the left? Yeah, so this picture here, the left on the top, is what we call epithelial tissue. Epithelial tissue. So epithelial tissue, the best way to remember it is that it's covering and lining tissue, covering and lining tissue, meaning it covers the outside of your body and it lines the inside of your body. Hey, who remembers from last week? I told you what my favorite type or my favorite organ system was. Does anyone remember which one was my favorite? Which one does Dr. Allis love? Yeah, Kira's right. I'm working on teaching my daughter that too. She's getting there to know that it's my favorite. Digestive system is my favorite. In the very middle of the digestive system, right next to your food, we have epithelial tissue. But epithelial tissue is also what we have on your skin, what you see when you look at your hands, when you look at your face in the mirror. That's epithelial tissue. So lining the very inside of you, lining the very outside of you, or covering the outside of you, that's epithelial tissue. With epithelial tissue, I kind of mentioned this before, but I'll reiterate that this type of tissue is all cells. It's all cells. So these dark spots that you're seeing here, these pink spots, these are the nuclei of these cells, but all of this is cells stacked on top of each other. So we'll talk about how to name epithelial tissue here in just a moment. But when you look at epithelial tissue, you're not going to have space for this extracellular matrix stuff, all those proteins on the outside. It's just going to be all cells. All of this is cells. That's epithelial tissue. My last type of tissue over here, can anyone tell me its name? What's the last kind of tissue that we're seeing over here? Who's this? Yeah, this last type of tissue that we're seeing a picture of is nervous tissue. Absolutely, nervous tissue. When we talk about nervous tissue is going to kind of do all the things that make stuff happen in your body. So nervous tissue is your thinking tissue. It's your tissue that collects information from the world around you, figures out what to do with it. There are two types of cells that we see inside nervous tissue. There's these big ones that I see here. These big ones are the ones that actually do the thinking. We call them neurons. And then notice all of these little tiny spots. I'm circling where some of them are. Those little tiny spots that you're seeing are things called neuroglia. And those are the helper cells, neuroglia. They help neurons do their job. So neurons are the thinkers, neuroglia are the support cells, the one that keeps them alive. Nervous tissue in general, though, that's responsible for collecting information, responding to information, all that kind of stuff. I will mention for you that we are looking at pretty textbook type images of these four types of tissue. We also have a ton of images for you of these types of tissues in the student Google Drive. So when you log into the student Google Drive, which remember is in the lab resources area of Blackboard, you can click on that link to it. There is a folder in the student Google Drive that says tissue images from our slides. 110% recommend that you go through after class today, and especially in the next couple of weeks for lab, you start looking at those pictures, because those are pictures taken from our actual slides that we have in the lab classroom. Those are the kinds of pictures that you're going to see on the homework assignments, and those are the pictures you're going to see on the midterm exam. So again, tissue images from our slides. You can start practicing looking at what actual connective tissue looks like, not just the super pretty, they have it in a textbook kind of picture. So the four main types of tissue, make sure we can tell them apart from each other. Let's talk more specifically about this one right here. Let's hone in on epithelial tissue. When we talk about epithelial tissue, there are two ways that we name it. We can either name it based on the number of layers that we have of the cells, or we can name it based on the shape of the cells. So let's start by talking about the shape of the cells. When I look at epithelial tissues, there's kind of four main shapes that I use for them. The first shape that I'm seeing right here is a cell that's about as wide as it is tall, or it kind of looks like a cube. Yeah, so a couple of us have chimed in here. When we see an epithelial cell that kind of looks like a cube, its technical name is cuboidal. That's really easy, right? Cuboidal epithelium looks like cubes. Now, it may not always look as square as that picture looks. Sometimes when we're looking at actual cuboidal epithelium, it's still going to be a circle, but your big tip-off that you're looking at cuboidal epithelium is that its nucleus in the middle is going to be really large. So a cuboidal cell is about as long as it is tall, so about as wide as it is tall. It's kind of cube-shaped. It's going to have a really big dark nucleus in the very middle. Let's compare a cuboidal cell to the type of cell we see right here. This is a shape called squamous, or you might hear it called squamous. Exactly the same. You can use either word for it. When we talk about squamous epithelium, this kind of epithelium is really squished. So we've got a cell that's really long and skinny. The middle part of it, where the nucleus is, that's the thickest part of the cell, but at the two edges, it's going to be really flat. So when we start looking at this type of tissue on slides, when it's not a model, it might look something like this. Or even when we're looking at it on actual tissues too, you might just see kind of a flat line of cells. So squamous epithelium is really flat. It's really squished. Cuboidal epithelium, about as wide as it is tall, or kind of cube-shaped. And then we come over here, and I'm looking at some cells that are a whole lot taller than they are wide. They're kind of column-shaped. What's the name of this shape of my last kind of cells over here? The ones that are column-shaped. Yeah, several of us are chiming in here. Those ones are called columnar cells. Columnar cells. So they are about as wide as they are tall. Or excuse me, they're way taller than they are wide. Dr. Alice, come on now. We're about as wide as we are tall. That's columnar cells. So when we look at epithelial tissue, the first thing you want to figure out, what's the shape of the cells? Are they tall and skinny? Are they flat and squished? Or are they shaped like a cube? That's the first thing we want to consider. The second thing we want to consider is how many layers that we have of them. So when we look at our types of tissue, the ones we've looked at so far are what we would call simple epithelium. Simple means that there's one layer, simple epithelium. And when you're trying to figure out if you have one layer or multiple layers, you're going to look at the location of the nuclei. You're going to look at the spots. So see how each of these spots, each of these nuclei are all in one line. There's one line of cells. Same thing over here. I've got one line of nuclei in these cells and one line of nuclei. Simple epithelium, whether it's tall and skinny, whether it's short and stout, if you will, or whether it's flat and squished. If there's just one row of cells, we call that simple epithelium. If there's more than one row of cells, we call that stratified. So stratified means that there's multiple layers. Like we're seeing here with stratified cuboidal epithelium, maybe there's just two layers. Stratified just means there's more than one, so maybe there's just two. Or maybe when we're looking at stratified squamous epithelium, maybe there's tons and tons of layers. In a few weeks when we start talking about the skin, we'll talk about how there are places in your body where we like literally have 50 layers of cells. So stratified squamous epithelium in places like the palms of your hands or the soles of your feet, there's literally 50 layers of cells, all kinds of cells there. So stratified can be as many as 50, it can be as few as 2, but it just means there's more than one. Here's a word that Dr. Alice likes here. Pseudo-stratified. Pseudo-stratified. This beginning part right here, pseudo, means fake. So whenever you see pseudo-stratified, you should be thinking fake stratified or fake more than one layer. There's only one kind of cell, one shape of cell that does this pseudo-stratified pattern, and that's our cells that are columnar cells. So the ones that are really tall and skinny, they do pseudo-stratified columnar. Pseudo-stratified columnar means we're pretending like there's more than one layer of cells, but actually there's just one. So if we looked at each of these cells, even though their nuclei are kind of at different places, each of these cells goes all the way down and touches the bottom down here. This bottom part, by the way, I'll give you a word for it. This bottom part is called the basement membrane. If all of the cells touch the basement membrane, it's pseudo-stratified columnar epithelium. If all of the cells have their nuclei in a straight line and they're all touching, that's simple. But because these nuclei are all over the place, we are pretending to be more than one layer. We are pseudo-stratified. There's one other arrangement of tissue. We'll talk more about this one next week. It's a type of epithelium called transitional epithelium. And the challenge with transitional epithelium is that the shape of the cells in transitional epithelium changes. So sometimes they look kind of cuboidal, sometimes they look squamous, they're going back and forth and changing shapes. So transitional epithelium is kind of its own thing. We're really not going to focus on that with our naming patterns, but know that there's this other kind, we'll see it next week, called transitional. It does have more than one layer, but because these shapes are constantly changing, we just call it transitional. I've talked way too much. How do we feel about the shape words? How do we feel about the layer words? Thumbs up, thumbs down. What questions do we have? Shapes and layers. Perfect. Okay. So you will see some questions on this week's lab homework assignment asking you to apply these words. Simple always means one layer. Stratified always means more than one layer. Those shape words, use what the shape of the cells looks like to tell you. I should also mention too, when we're talking about a stratified type of epithelium, when we're talking about something with more than one layer, make sure that you always look at the top layer, what we call the apical layer. That's the anatomy word for top. Always look at that apical layer to name the shape of the cells. So we never want to look down here by the basement membrane because that'll get us all worried and freaked out. We'll say the wrong shape. Always look at the apical layer, the one at the very top. That's what we do when we have stratified epithelium. Let's talk a little bit more about some of the structures you might find on the apical surface or the top surface of epithelial cells. Now, in particular, we are only talking about the types of cells that are really tall and skinny, or tall and skinny cells. What was the name of my cell shape that was tall and skinny again? What's those tall and skinny cells? Tall and skinny. Yeah, those are my columnar cells. Columnar cells. Okay, when we talk about tall and skinny columnar cells, there are a couple of different things they can have on the top of them. One of the things they can have on the top of them are what we see over here. This is an actual tissue slide that you're seeing here. So all of these little things on the top that look like eyelashes, these are cilia. So one of the things that our columnar cells can have on the top of them are cilia. And cilia look a lot like eyelashes. Eyelashes. The other thing that columnar cells might have on the top of them, though, is what we see on this picture. And these are things called microvilli. Microvilli. And when you're looking at tissue that has microvilli in it, this is going to look a little bit more like peach fuzz. A little bit more like peach fuzz. So notice how I've got these long, skinny eyelashes up here. These are my cilia, versus that peach fuzz that I see on the top of these cells here. These long cilia, these eyelashes, are really good at moving stuff. So we find them in places like the trachea, where we want to move out any dust or debris that you might accidentally breathe in to make sure it doesn't get down to your lungs. Cilia, great at moving things. Microvilli are actually just places where the plasma membrane of these cells folded into lots of little tiny fingers. Because I folded the plasma membrane into these little fingers, that means I have more space where I can touch your food. Notice how we're in the small intestine here. The small intestine is the part of your digestive system that absorbs the most nutrients. So I'm going to see these places where the membrane folded a whole bunch to make these microvilli in places where I want to absorb a lot of nutrients. So this peach fuzz brush border up here, if you will, these are microvilli. Microvilli really good at absorbing nutrients. Cilia, these eyelashes, really good at moving things. So either moving stuff out of the throat, or we see them as well in the fallopian tubes, taking an egg from where the ovary releases it down into the uterus. Cilia, all about movement. Microvilli, all about absorption. I believe, let me check my slides here. Yeah, I believe that is everything I wanted to chat with you all about with those 4 main tissue types. Before we go back to the cell cycle and mitosis, do we have any tissue questions before we back up? I'll do a Monsters, Inc. quote here. Your stunned silence is very reassuring. Anybody know my quote? Has anyone seen Monsters, Inc.? I'm getting older and older. I don't feel like it, but apparently I am. Okay, I got a few of us that like it. Exactly. Yes, it's all about Mike forgetting to file his paperwork. Absolutely. Yep. All right. Well, if I have no protests from everyone, I'm going to go ahead then and we'll transition back to lesson number 3. Yeah, now we're tossing out all of the great quotes from Monsters, Inc. I wonder, let me see. Oh, the monster emojis are very lacking. That's as close as I can get. It's a little alien, but we got some Monsters, Inc. going on in the chat. That's perfect. All right, so let's step back then, and let's talk about cells when they need to divide. So you are constantly in your body building new cells. Some of those cells, when you're younger, are helping you to grow, make you taller, make you stronger. Some of those cells, when we're to the ages where we all are now, are replacing cells that are old and worn out. So the process that a cell uses to divide itself, to make a copy of itself, whether it's for building or for repairing your body, it's always going to go through the same sets of steps to do that. Let's talk the 2 words that are important to cell division. There's 2 words that are important. The first word that's important is something called mitosis. The second word that's important is something called cytokinesis. Cytokinesis. I want to make sure before we dive into any of this cell division stuff that we know at the beginning what the difference between mitosis and cytokinesis is. A cell has to do both of these things to go from being one cell to being two cells. But these two processes divide different parts of the cell. Does anyone happen to know from your working before class today, what is a cell dividing up when it does mitosis? What is it splitting up when it does mitosis? Any ideas? Yeah, exactly. When we talk about mitosis, mitosis is all about dividing the cell's DNA or dividing its genetic information. Absolutely. Half of the battle of going from one cell to two cells is both of those cells need to have the DNA or need to have the directions to be able to live. So I got to divide up those directions. I have to divide them up equally, correctly. That's what mitosis does. But the other thing that I have to divide when it's time for my cell to go from being 1 to 2 is I need to divide up the cytoplasm. The cytoplasm. The process of cytokinesis is when I divide up the part of the cell called the cytoplasm. Now, remember from our lecture discussions, the cytoplasm has two parts, or there's two things that make up the cytoplasm. Does anyone happen to remember what those two things are? Two things that make up the cytoplasm. One of them looks a whole lot like that word. Yeah, so one of the things that the cytoplasm is it's the soup inside your cells called the cytosol. So part of the cytoplasm is the soupy stuff. The other part of the cytoplasm are the organelles. The organelles. So the cytoplasm of a cell is the soupy stuff inside of it and the organelles. Things like the mitochondria, the ribosomes, the rough ER. When a cell goes from being 1 to being 2, we go through a breakup, we got to divide up the genetic information, we've got to divide up the soup and all the organelles that are inside of it. Mitosis and cytokinesis. We need both of them for a cell to correctly divide. So let's start with a reminder here of the genetic information of the cell. I'm looking at the genetic information of the cell when we're getting ready to do mitosis. We're getting ready to split it up. When we talk about this X-shaped form of the DNA again, what was the name of this X-shaped form of the DNA? This whole thing that we're looking at, my whole model here, the whole thing, yeah, absolutely, is called a chromosome. This entire model that we're looking at is a chromosome. A chromosome, this X form, is actually going to be split in half, and we'll see that as we look at the process of mitosis. We're actually going to chop it straight down the middle. So an X-shaped chromosome is actually made of two halves that are exactly identical to each other. So these two halves, each half of a chromosome, the special name for that, I see it popping up in my chat here, is what we call a chromatid. A chromatid. A chromatid is an underline highlight star word because I want to make sure we don't confuse chromatid with chromatin. Chromatin. We already used that word chromatin today. What was up with chromatin? What is chromatin? Do we remember? Chromatin. I know I'm making you type a lot. Yeah, absolutely. Chromatin is that looser form of DNA. Absolutely. The spaghetti noodle mess form of DNA that would be really hard to divide. Chromosomes, easy to divide because they're packaged. We've packaged that DNA into two small structures called chromatids. I can split those chromatids apart, and now everybody's got genetic information. Chromatin, it's going to be real hard to split that spaghetti noodle mess equally between two cells. So chromatin in the nucleus, what a cell uses when it wants to build proteins. Chromatids, when a cell's getting ready to divide, it winds up those spaghetti noodles, turns them into this X-shaped chromosome. The way that I attach these two chromatids to each other is using a structure here in the very center of a chromosome. This structure here is called a centromere. A centromere. And the centromeres are going to be really important when we're talking about dividing up the genetic information, because we're going to attach spindle proteins or some divider proteins to the centromere. They're going to play tug of war with this chromosome until there's so much pressure on the centromere here in the middle that it literally breaks in half, and these two chromatids go to opposite sides of the cell. So X-shaped chromosomes held together by a centromere in the middle. Chromatids are each half of the X. And this last area, by process of elimination, when we look up close at what's going on here in my chromatid, we've got all of these little sections of DNA really tightly folded up on each other. That's what this last area represents, that DNA. When we talk about these X-shaped DNA, when we're looking at that chromosome from our last slide there, the only time in a cell's life cycle, so the only time from the beginning of its life when it's born until it divides into new cells, the only time you're going to see X's is the part here in yellow. Most of the time in all these parts here of the cell cycle that's represented as green, most of a cell's lifetime, its DNA is in that loose form called chromatin. Most of the time, the cell's DNA is in chromatin. Now, here is some important terminology for me to emphasize with you all. This word right here, interphase. When we say that a cell is in interphase, this is the part of the cell cycle from when it's first born, if you will, until the cell gets completely ready and starts to divide. Interphase is what a cell spends most of its life doing. So this is the cell growing up, if you will. So let's put some notes here. Interphase. Interphase is a part of the cell cycle, part of the cell's lifespan. When a cell is in interphase, it's doing things like our picture shows us, like growing. So that cell is getting bigger. It's doing things like copying its DNA. It's doing things like building new organelles. Interphase is all about that cell getting ready to divide. Interphase is, like I said, a part of the cell cycle. That's different because then we go into mitosis. Mitosis. And we already talked about this. You all already told me. When we talk about mitosis, what is it that we're doing in mitosis? What happens in mitosis? Yeah, exactly. When we enter the process of mitosis, we're dividing the DNA. Absolutely, dividing the DNA. Mitosis takes place in 4 phases. So 4 phases of mitosis. So I'm going to type them real fast. You can see them on the picture. They're prophase, metaphase, anaphase, and telophase. The 4 phases of mitosis. I emphasize this with you because you're going to see homework questions that will say this part of the cell cycle is when something occurs, or this phase of mitosis is when something occurs. When I ask you about phases of mitosis, you only have 4 options: prophase, metaphase, anaphase, telophase. If I'm asking you about a part of the cell cycle, we only have 2 options. We talked about one of them, which is interphase. The other option for part of the cell cycle is cytokinesis. Cytokinesis is another part of the cell cycle. When I talk about cytokinesis, what did we say was happening again? Do we remember what happens in cytokinesis? Yeah, exactly, Kira. Cytokinesis, we divide the cytoplasm. When we're splitting up the soup, when we're splitting up the organelles, that is its own thing. That's cytokinesis. When we're splitting up the DNA, dropping those chromosomes, pulling them apart, all that jazz, that's mitosis. Please don't tell me when you have a homework question, if the question asks you about the part of the cell cycle where we pinch the plasma membranes to make 2 new cells, please don't tell me one of my phases of mitosis. My phases of mitosis, I'm going to ask about specifically using this terminology, the phases of mitosis. When I ask about the parts of the cell cycle, now we're talking about either interphase, where my cell is growing, where it's copying its DNA, where it's making new organelles, or the very end, where it's splitting up its soupy stuff. Cytokinesis. Those are my 2 parts of the cell cycle. My 4 phases of mitosis. Yeah, Audrey mentioned that was a little tricky for her on the Blackboard assignment. Absolutely, yes. So for any friends who haven't tried yet, please, 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 I can't emphasize this enough, look for this terminology. Am I asking you for the part of the cell cycle or the phase of mitosis? Once you start working on the assignment, you'll understand why I am saying these things, because there are lots of questions like that. So look for that terminology, phase of mitosis or part of the cell cycle. Yeah, Kaylin's mind is blown. It'll save you a lot of frustration now that we know that. What I'm going to do with you now is I'm going to walk you through some super pretty pictures of what each of these phases of mitosis look like. Then I'm going to show you the kinds of slides that we use in our lab class to look at the phases of mitosis. We're going to do our best to use the time that we have left to get good at identifying the phases of mitosis on our slides. So I'm going to show you and explain what's happening in all these beautiful pictures. Our slides aren't nearly this beautiful, but we're going to try to come up with some hints or some tips for us to help us with those slides. As I talk about these things, what I would recommend that you do is pull out a blank piece of paper and take some notes about what I'm telling you is the most important thing that happens in these phases, or what I'm telling you to look for in each of these phases, because that's going to help us out a lot when we start trying to identify them. So maybe put your lab packets aside, just grab a piece of paper. Let's talk about what happens in each of these parts of the cell cycle or these phases of mitosis. This first image that you're looking at right here, this is a cell in interphase. So this is a cell that's growing, that is getting bigger, that's eventually going to work toward the process of dividing. When I look at this cell in interphase, let's talk about our colors here. Blue is representing the genetic information in this cell. Notice when a cell is in interphase, all of that genetic information is here in the nucleus, and it's in that really loose form, that spaghetti noodle mess form. What did I call my DNA when it's in the nucleus and it's really loose and fluffy? What kind of DNA do we see here in blue? Yeah, Kira's right. There we go. A couple more of us chiming in here. This blue, solid blue mess that I see here, this is the chromatin. This is the genetic information that is available for your cell to use it to make proteins. Absolutely right. Outside of the nucleus, notice this little green thing that I see right here. These green things are the centrioles that we identified before. So they've stained them green in this particular cell. And then the red that you're seeing out here are all of the regular proteins that we find, something called the cytoskeleton of the cell. What we're going to want to pay attention to as we start looking at the different phases of mitosis is the blue color and the green color, because that's going to tell me what's happening with the DNA and what are those centrioles doing. So when you look at a slide and you have a cell that's in interphase, in our cells, you may not actually even see the nucleus of a cell that's in interphase because the genetic information in that cell is not condensed. It's not going to maybe be as dark in color. If you do see a nucleus in a cell that's in interphase, it's pretty much all going to kind of be the same dark purple color inside of it. You're not going to see any chromosomes. So it's all going to kind of be the same dark patchy color. Once I start mitosis, once I go into the first phase called prophase, the first thing you're going to notice in prophase is that the cell takes its spaghetti noodle mess, that's all blurry here, and starts to condense it down into chromosomes. So the first thing that happens in prophase, they're calling this early prophase. The first thing that happens in prophase is I form chromosomes. So when you're looking at a cell that has started prophase, see how this nucleus looks a little bit spotty? I'm starting to see the beginnings of chromosomes. I'm taking that spaghetti noodle mess and I'm turning it into packed forms of DNA. In early prophase, we first start to form chromosomes. By the end of prophase, not only do I have all of these chromosomes that I can see here, but also at the end of prophase, I lose the nucleus. So when you're looking at a cell, if you see black squiggly lines that are kind of all over the place, there's no rhyme or reason to them, they're probably in prophase, because in prophase, that nucleus falls apart. So here's what happens for your notes purposes of what's going on here. In prophase, first thing that happens in prophase, I form chromosomes. Second thing that happens in prophase, the nuclear envelope or the nuclear membrane dissolves. By the end of prophase, you've got a bunch of chromosomes floating around in the cell, kind of haphazardly, not arranged very well. After prophase, my first phase of mitosis, we move into metaphase. Metaphase. And this one should be the easiest for you to identify. Metaphase, because in metaphase we form this thing called the metaphase plate. So the metaphase plate is when all the chromosomes line up in the middle of the cell, because we have a little bit of tug of war going on here. This is where one centriole is up at the top. Here's where another centriole is here down at the bottom. Notice all of these spindle proteins, the mitotic spindle that's shown here in green. They attach to each of the chromosomes at the centromere, and then we pull a little bit this way, we pull a little bit this way. Pull back and forth, back and forth, back and forth until everybody's lined up in the middle. And when we're in the middle, when we meet in the middle, that's metaphase. So when all the chromosomes are in a line in the middle of the cell, that's metaphase. Metaphase can only last for so long before, so here's where we were before, here's metaphase. This can only last so long before those centromeres fall apart. The strain from pulling left to right breaks them. That brings us into the next phase of mitosis called anaphase. In anaphase, you'll actually see two groups of chromosomes that are moving to opposite ends of the cell. So here's one of my centromeres. Here's the other centrioles. One centriole up top, one centriole down below. Notice how I've got little stringy chromosomes that are going toward the middle of the cell on both sides, because I literally just split those X's in half, and now I'm pulling them to opposite sides of the cells. So in anaphase, you're going to see two groups of chromosomes being pulled to opposite ends of the cell, anaphase. The last phase of mitosis is called telophase. Now notice that on my picture here, they've got telophase and cytokinesis labeled in the same picture. Here's where that terminology is really important. Remember that cytokinesis was part of the cell cycle, so it's not technically part of mitosis. But this last part of mitosis, that's telophase, happens at the same time as cytokinesis happens. So because of that, the way you're going to identify a cell in telophase might actually have a lot to do with some of the cytokinesis stuff. Let's start with what actually happens in telophase. What actually happens in telophase is I form two new nuclei, so two new places in my cell where my genetic information is being stored. Notice how when you look at this blue circle right here and this blue circle right here, this blue part right here, see how these look a lot fluffier compared to these little stringy lines that we see over here? By the time we get to telophase, the last phase of mitosis, my genetic information actually starts to become chromatin again. It gets looser again. So when you're looking at telophase, you're going to see two looser parts of genetic information that are completely separated from each other. That's my biggest difference, because in telophase we form new membranes and we make chromatin again. Cytokinesis was when we divided up the cytoplasm, so all the soupy stuff, all the organelles. The biggest thing, the biggest tip-off for cytokinesis is we form something called the cleavage furrow. I know it's kind of small there, but that terminology, the cleavage furrow. This is, see how there's kind of a divot right here in my plasma membrane, and there's a divot right here. Literally what we're doing when we divide the cell in cytokinesis, think about putting one finger right here and one finger right here and pinching them together. That's literally the way we divide the membrane. So cytokinesis, we pinch one side to the other side to make two new cells. That pinching forms something called the cleavage furrow. So the cleavage furrow shows me that cytokinesis is happening, that we're dividing up the cytoplasm. When we divide up the cytoplasm, that happens around the same time as telophase. So around the same time as my DNA starts to get loose, that's when I start to divide up that genetic information as well. Help me out in the chat. I think you have these pictures in the guided lesson for number 3. Is that correct? The online guided lesson has these pictures? Okay, so a couple of us are saying we've seen them before. I bet that's where you've seen them. So I'm going to move on from these pictures because they don't actually match our lab pictures, but you can see them again and reference them in the lab guided lesson for lesson number 3. Our pictures are going to look a little bit more like this. Oops, too far. When we are looking at the phases of mitosis on the slide that we would use in our class, so if we were in person and we were looking at the phases of mitosis, we would use something called the fish Blastodisc slide. Let me write that for you. Fish Blastodisc. This means we've got a bunch of cells that are dividing and growing. Our cells don't have the beautiful blue color and red and green. What we do see with our cells is that the genetic information looks darker. So I'm seeing some dark purple that's representing that genetic information. The cells overall are kind of pink. So when we look at our cells and we're doing the process looking for the phases of mitosis, keep in mind that we're not going to have the fun blue and red and green. This is what we got to work with. When I'm talking about the phases of mitosis, let's remind ourselves of what we said happened in each of them. So we start mitosis with what's called prophase. Can you help me out in the chat? There were two things that I told you happened in prophase. Two different things that happened in prophase. Do we remember what those things were? Okay, so one of them Kira's honed in on for us. Chromosomes form, absolutely. That's the first thing that happens. We build those chromosomes. Yeah, and then the second thing that happens is that we lose the nucleus. Absolutely. Two things that we're looking for in a cell that's in prophase. We form our chromosomes, we lose the nucleus. So check this out. I've got a cell. Here's my whole cell right here. I've got a cell. Notice how there's some dark spots in the middle of this cell and how it kind of looks like there's not really a line or something that's holding them all together in place. This cell doesn't have a nucleus anymore, and we've started to form some chromosomes. That's why it looks dark and kind of spotty, because I've started to form chromosomes. This is an example of a cell in prophase. Yeah, and exactly like Alapina mentioned, this one would be considered late prophase. I'm not going to ask you on the assignment, is it late or early? Just know that this is what prophase looks like. I can see some chromosomes forming, and I don't have a nucleus anymore. That's prophase. Remember I told you metaphase is probably going to be the easiest to recognize. In metaphase, my chromosomes meet in a line. So here's that line of chromosomes. Here's actually one of my centrioles I can see over here. See the little fibers or little proteins that attach to those chromosomes in the middle? So my other centriole is missing, but you can see some of those spindle proteins that are there. Metaphase, those chromosomes line up. They meet in the middle. In anaphase, remember that the chromosomes split into two different groups. Notice how you can kind of see some of those dark lines that are pointed toward the middle of the cell, because these genetic information clumps are still dividing. We're still pulling these ones over here. We're still pulling these ones over here. So their genetic information is trailing to the other side of the cell. This is anaphase, where the chromosomes are being split from one another. The last phase is telophase. Remember in telophase that I told you we have two distinct groups of genetic information. Notice how this one's gotten fuzzy. This genetic information is becoming chromatin again. This one hasn't quite started that yet. But the way I know this is telophase and not anaphase is because of that structure we talked about, that cytokinesis structure we talked about. This divot right here and this divot right here. See how the membrane comes in? The membrane is pinching in on the two sides. I can tell it's telophase because I have that thing called the cleavage furrow, where I've started to do cytokinesis. I'm dividing up the soupy stuff. I'm pinching in the membrane. So this last one here is telophase. What phase of mitosis is telophase? Because I can see the cleavage furrow on these two sides here. I know this was a super crash course in mitosis, and mitosis is going to take a lot of practice. Here's how I want us to start that practice in the last bit of time that we have together. I'm going to split us up into groups one more time. Here is, if you go back into that same thing I shared with you before, the very last slide has this picture. I want you to work with your groups on identifying which phases of mitosis we see in these pictures. So I'm going to split us up into groups. I'll give us again about 5 minutes to see if we can identify things. You might use the phases more than once, so don't be afraid if you're using something more than once. You might do that. For my friends watching via recording, this we will not be able to go over this together in the recording because it's going to stop when I split everyone into groups. But feel free to reach out to your friends who are in the class session today to get our answers to this. So I'm going to split us back into groups one more time. Once you're in that group, pull up your slides. It's the very last one in that group in your slideshow. Let's work together to see if we can practice identifying some of these phases of mitosis.